Good day for everyone. Now, today we will learn something about the biotechnology in animal production and also the animal reproduction. So, biotechnology in livestock improvement is used as a tool since years, and it is one of the important tools for the development of the livestock breeds and also certain production and also reproduction. So, the biotechnology provides one of the new tools for improving the human health and animal health with welfare and increasing livestock productivity is also one of the important criteria for this particularly the livestock technology is concerned. Whereas the biotechnology improves the food we eat, meat, milk, and eggs. So, biotechnology is also useful in combating the disease that is occurring in case of the animals. And also, just like other assisted reproductive techniques such as artificial insemination is also one of the biotechnological tools, embryo transfer, in vitro fertilization, livestock cloning, and also other healthier springs and to produce more and more important things that is very much required to the human health or the animal health is concerned. This is the history of biotechnology starts in 1972 is concerned, and it is one of the episodic way in which the DNA composition of human showed to be 99 similar to that of the chimps and gorillas. So in 1997, genetically engineered bacteria are used to make human growth protein because earlier it was collected from the cadavers and it used to cause a lot of allergy, etc. But later this has been replaced by the genetic modification of certain things, especially the apart from this one, the North Carolina scientists they also found the Hutchison and Edgerly fruit possible to introduce specific mutations at specific sites in DNA molecule. 1979, the first monoclonal antibodies are synthesized. The US Supreme Court approves patenting genetically engineered life forms. The US patent for gene closing is awarded to Boyer and Cohen. Then 1981 is the first mice to be successfully cloned. Then the 1982, the human insulin drug produced by genetically engineered bacteria, that's the first biotechnology product approved by FDA. 1983, the first artificial chromosome is made. 1983, once again, the first genetic markers for specific inherited diseases are found. In 84, the DNA fingerprinting technique is developed. Then also the first genetically engineered vaccine is developed. In 1986, the first biotech-derived interferon drugs for the treatment of cancer are synthesized. In 1988, Congress funds the Human Genome Project, and microorganisms are used to clean up Exxon Valdez oil spill and many more. Then, so this goes. So, especially in drug discovery, etc., the biotechnological tools were introduced into the uh, very, very effectively. And especially the in 2000, the pigs are the next animal cloned by the researchers and to help produce organs of the human transplantation. The sequence of human genome is published in Science and Nature articles. So, 2002, the scientists completed the sequence of pathogen of rice, pathogen of rice fungus that ruins enough rice to feed 60 million people annually. Then Dolly in 2003, the first cloned ship is euthanized in 1996. It is euthanized just to find what are all the changes in that particular thing. And animal biotechnology is based on science of genetic engineering. Under the umbrella of genetic engineering exist other technologies such as transgenic and cloning that also use the animal biotechnology. So animal biotechnology has lot of potential to increase the growth rate, enhance lean muscle mass, enhance resistance to disease, improved use of dietary phosphorus to lessen environmental impacts of the animal manure. So the transgenic poultry, swine, goats, and cattle that generate large quantities of the human protein in eggs, milk, blood, and urine have been produced with the goal of using the products as human pharmaceuticals. The human pharmaceutical proteins include enzymes, clotting factors, albumin, and also some of the antibodies. Then the animal biotechnology has the main objective of characterization and identification of the animal breeds. And animal biotechnology has a widespread and diverse applications of the areas of food quality control, analysis of the milk produce, milk and other animal products, development of disease-free animals. And apart from this one, the animal biotechnology implies the application of scientific and engineering principles to the production and processing up of the materials. Genetically engineered animals can be used to develop new medical treatments. And the animal biotechnology or sometimes the recombinant DNA technology has advances in human health to improve the animal production and also the health care and welfare and enhancement of animal products, increased milk productivity, then also to environmental and conservation friendly benefits are there. So the applications are developed through the especially research in livestock, poultry, fish, insects, companion animals, and laboratory animals. So market applications developed through research have led to the emergence of three scientific and animal biotechnology sector. So animal genomics, number two is animal cloning, third one is the genetically engineered animals. So the animal models were developed especially in the research to combat certain diseases like the polio vaccine, cataract surgery technique, and also to dialysis. And the FDA also regulated several new drugs, medical procedures, and cosmetic products which must pass safety tests. Involves phase testing to conduct a significant number of trials on cultures, cell cultures in live animals and on human research participants. So the animals most often used are the mice and rats, zebrafish, fruit flies, nematode. So alternative to animal models are also cell culture and computer generated models were also used as the biotechnological tools. And animals in research, lot of institutional review boards like IASC, then Federal Animal Welfare Act and CCSA, etc. have been produced. So applications of the biotechnology in animal is the embryo transfer, then animal breeding, animal vaccine production, then animal nutrition also, then production of transgenic animals or knockout genes, etc. So, in breeding, recombinant DNA technology has been possible to breed animals with great precision and accuracy. And specific genes can be inserted into an animal embryo without causing a shift in other genes, which are present in the same animal, and also the twinning technology, etc. So, the milk of ordinary cows lactoferrin, an iron containing protein, which is significant for the infant growth. So this has been developed like transgenic bull human, which has been micro-injected with the human gene for lactoferrin, and the breeding human Herman and Herman, not a human or the Herman, and its progeny will improve to be a new source of nutritious milk. And applications, especially in case of the vaccinology, billions of dollars are spent on each year to improve upon farm animals and their healthcare. And scientists are now trying to use recombinant DNA technology to produce vaccines for the animal diseases. So the conventional vaccine production is very high cost and it produces low value, whereas recombinant vaccines are efficient and score on their fast pace of development. And the conventional vaccines can take as long as 20 to 30 years of research and experimenting before they are ready to use, whereas the modern vaccinations. So the modern vaccines are made readily in much shorter lifespan and are very potent. And the nutrition, some of the points need to be taken over is so giving small amounts of the proteins for the animals like sheep and cows has already shown to increase the animal feed conservation efficiency. Like the bovine somatotrophins can overexpress in bacteria and generated in larger quantities for commercial purpose. And the biotechnological manipulation can help to generate porcine somatotrophin, which is not only improves the feed efficiency by 15 to 20%, but also important benefits of the human system. PST also helps to reduce fat deposits, and also then the embryo transfer is another technique of genetic manipulation. The principal advantage of embryo transfer is that it increases the reproductive capacity of useful cattle like cows and buffaloes, even if they are not productive naturally. And transgenic animals, like they are the cloned animals, created waves all over the world, and this feat is indeed the significant and the clone of the donor. So the xenotransplantation means organ transplantation have proved to be a very cost-effective treatment for the heart, kidney, lung, and other diseases. So the first gene transplantation, like the organs taken over from the pigs, they what it was conducted in 1905. A French surgeon transplanted slices of the rabbit kidney into the human patient, and the transplanted the heart walls of pigs are commonly used to treat different forms of the severe heart diseases. So there are varieties of the terminologies are there. Of course, we are all very well aware of this cell generation time, then cell hybridization of few to two or more dissimilar cells leading to formation of the synkaryon. A cell line arises from the primary cell culture. Cell strain cloning means it is producing the copy of one animal in a very similar pattern, and continuous cell culture is also one of the technique, and cryopreservation is one of the technique. Already it is used in the artificial insemination technologies. And hybridoma, the cells results from fusion of antibody producing tumor myeloma and antigenically stimulated normal plasma cells. And Many more terminologies are there here: subculture, potency, transfection, and many more. And it is the, once again, to recall some of the things, it is the science of altering genetic and reproductive process in plants and animals. And aim of biotechnology in livestock improvement is to increase genetic variation, increase accuracy of selection, reduce generation interval, increase selection intensity. Then by manipulation of the gene, without manipulation of the gene also it is produced. So multiple ovulation and embryo transfer technique can be used, and also the artificial insemination, oocyte harvesting, in vitro oocyte maturation, in vitro fertilization, nuclear transfer and embryo cloning, and also the sex determined semen production. So the artificial insemination is one of the oldest technique. Of course, it is has got the dramatic improvement in the population of the cattle, which made India to be number one in world in the milk production. Then the multiple ovulation or embryo transfer is also used in case of the animals for growing the embryos and producing the good quality embryos and transfer. And this technique is also used in case of the human being, especially in infertility problem. So here the superovulation can be done with the gonadotrophic injection. Then artificial insemination is done with to initial superovulation, then harvesting up of the embryos 6 to 8 days or for inseminations, then the isolation and purification of the embryos, then storage of the embryos or preservation in liquid nitrogen at uh, or at 37 for temperature for 1 days. Then transfer of the embryos into the recipient surgically or non-surgically. Nowadays, non-surgically, a catheter can easily be passed and palpated. And sometimes the ultrasound guided things are always very popular. And the birth at 9 months of embryo transfer will happen. Then oocyte harvesting or OPU, in vitro oocyte fertilization or maturation, is taking place, and it has already gone several type of the researchers and development, and the OPU can be applied to pregnant animals as well as the pre-pubic animals. The impact of these methodologies and genetic response operates through the same channel as MOET and increase the selection intensity of the female side and increase the selection accuracy of the male and female side also. So this technology is also used in case of small and large ruminants, especially oocyte maturation, in vitro fertilization, then embryo transfer to a female seed. Then nuclear transfer of embryo cloning is also there. So this type of the in the recent technologies have been very well adopted by the scientific community, by the organism to be cloned. Some of the cells will be taken, then they will be subjected for developing an oocyte, or it is called as the embryonic stem cell. So the unfertilized egg cell will be transferred to a glass pipette smaller in a diameter than the hair. Then egg cell with no nucleus and egg cell and cell to be cloned are fused together. Then they are implanted into the EU. So that's the easy technology nowadays. So sex selection nowadays, the based on PCR and other thing, and also the X or Y chromosomes are being separated, and sex determined semen is produced very commonly, and also many of these techniques are very commonly implemented. Gamete and embryo cryopreservation is also taken place in cryopreservation is already implemented in artificial insemination technique itself. And livestock genomics and marker-assisted selection is the first step in the process referred to as positional cloning, which culminates in the isolation of the casual gene and mutation. So all these things have developed, marker assisted, etc. Nowadays, as the previous classes also we have learned the animal biotechnology as is one of the growing area. Then genetic engineering means changing the already happening genes like the recombinant DNA or the they are deoxyribose nucleic acid was taking in the tiny bit of DNA containing the desired gene from one organism and splicing into the DNA strand of another thing. That is called as genetically engineering. So by this particular technology, many type of the vaccines are also being produced, and many type of the medicines are also developed. See, one of the medicine that's developed is the bovine somatotrophin or bovine growth hormone are proteins that affect the utilization of energy in the body more and more. Of course, the permission is not given in India for RBST, whereas in other countries it is there and causes energy deprived from the feed to be used in milk production rather than weight gain. Hence, the it causes energy derived from feed to be used for milk production rather than weight gain. So more and more milk can be produced. Previously, the only source of BST for research has been from pituitary glands of the dead cows, but nowadays the recombinant bovine somatotrophin can be produced. And future challenges are also there. The cost is very high. The ethical constraints are also there. Some of the world they oppose the biotechnology, and also major problem in fear of biotechnology by genetic public, because the people have a lot of misconception about genetic engineering technology. So there is scarcity of the properly educated scientists and technicians and lack of support for projects aimed at applying the biotechnological tools is also another thing which is a constraint factor. Then techniques, of course, artificial insemination, in vivo derived embryos, and IVF, in vitro fertilization techniques are already developed in many of the taken. And embryo transfer is already taken care, and 1,97,90,000 annually embryos are produced in the animals, especially in case of the genetically engineered substances. And the disease control and multi-benefit transboundary movement of livestock and all these things can be done. So embryo transfer is one of the better technology. Nuclear transfer of the cloning, multiplication of desired traits, conservation of the animal germplasm, and desire assist. So vaccination. Now most of the vaccinations or vaccines are produced from the biotechnological tool instead of the other traditional method of the production of the vaccine. Whereas the risk assessment is also there to identify the genetically engineered and the cancer genes etc. are already marked. Then, regulatory framework issues to be considered: environmental impact, food safety, animal health, trade, and many times, most of the time, it is ethical constraints are much more, and that's why the all these different type of the consideration. And it requires a sound scientific basis to evaluate the associated risks and future dangerous things, if any, has to be already elucidated. So this technology also evaluated to the nanotechnology. So the nanotechnology is the one where in which the research and development aimed at understanding and working with seeing, measuring, and manipulating matter at the atomic and molecular and supramolecular levels. So it correlates the length scales of roughly 1 to 100 nanometers. So that's called as the nanoparticle. At this scale, the physical, chemical, and biological properties of the material may differ fundamentally and often unexpectedly from those of corresponding bulk material. So the virus usually measures 75 to 100 nanometer. Proteins, they are in the range of 5 to 50 nanometer. DNA has got less than 2 nanometers. So the nanotechnology is used in animal health and smart drug delivery system, targeted drug delivery, etc. And early disease diagnosis and treatment is also possible. And animal breeding or real-time measurement of the blood disturbance DL changes can also be used using the nanotechnology. But there is a ethics or ethical definition is the qualities or characterize and motivate good scientists do not necessarily have any bearing on the ethical or sociological sequelae of the discovery. So, a practical framework for ethical assessment of the GM animals, is also another important type. Whereas the embryo transfer technology is one of the most adopted technology in case of the cattle. So the embryo transfer is one step in process of removing one or more embryos from reproductive trap of the donor female and transferring them to one more recipient females. So the embryos can also be produced in the laboratory via techniques such as in vitro fertilization or somatic cell cloning, whereas proliferation of so-called desirable phenotypes and production of artificial insemination bulls through planned mating is also that. So it is necessary for achieving success in various assisted reproductive technologies, especially in case of in vitro fertilization and the animal cloning. So embryo transfer was first performed, recorded by Walter Hilton in 1890. He transferred two Angora rabbit embryos into a distilling wiggly dough. So this was successful embryo transfer, and later it is reported in cattle and pigs by different scientists. Whereas the first commercial embryo transfer were done in early 1970s. Initially, embryos were recovered from valuable donors and transferred to the recipient animals using the invasive surgical technique. But now, non-surgical methods using the ultrasonographic guided things have been very much developed. Now, it is the principle is collect the embryo from elite animals, females, and transferring harvested embryo into the females of lesser genetic merit or renting the womb of that particular animals. And therefore, to achieve this goal, the bovine female ovulates multiple natural and matured and oocytes, which are capable of being fertilized in vivo and which can continue to develop as the embryos. And the equipments required are the binocular microscope, then wide tubing, then catheter and stilate with controller rated freezer, then also the filters, homostatic pads, and incisors are required. Then this is the technique as already it is explained. And the selection and management of donor and recipient is important. Genetically superior animals that contribute to the genetic objectives of the program and likelihood of producing large number of usable embryos are two broad criteria for selecting donor curves. So the donors are evaluated by rectal or vaginoscopic examination. Then the fertile bulls and fertile semen, which makes it especially important to use high quality semen. And donors are located either at the farm under the production conditions or at an embryo transfer center, frequently under intensive managemental system. And selection of management of the recipient is also another thing. And cows that are reproductively sound, exhibit calving case, and have got good milking and mothering ability are the recipient prospects. And proper plane of the nutrition is also very important thing. And proper recipient herd management is critical to embryo transfer technology success. And management of this herd requires fundamental understanding of the recipient, and hence we need to select. Then later the animals are synchronized the recipients by using PGF to alpha 12 to 12 hours of the recipient, and this requires synchronization of the estrus cycles between the donor and also the animals. Then the superovulation of the donor is taken place by making the animals to be stimulated with follicle stimulating hormone, and this hormone is administered twice daily for 4 days in the range of 8 to 14 days with functional corpus luteum at one ovary, and usually there will be a corpus luteum and also the this especially the follicle, and insemination of the curve will be done using the artificial insemination technique. Then correct AI techniques are used. Then this is the embryo recovery is there. Usually, in most cases, embryos are recovered 6 to 8 days after the beginning up of the estrus. Then embryos can be recovered non-surgically by 4 days after stress from some cows, but prior to 6 recovery rates lower than that of the 6 to 8. Then embryos can also be recovered on day 19 to 14 after stress, but it's always recommended to elongate dramatically or sometimes damaged during recovery and become elongated, especially after day 30. And the embryos, which other reason for choosing this time. So this is the embryo filter. Then later outlet is considered. Uterine flushing media is always put inside the cervix, etc. Then recovered. Then flushing will be done with the technologies. Then embryo handling and evaluation to be carefully done with the in vitro fertilization techniques with embryo transfer, sex determination, and cloning is concerned. Then in the laboratory, so under the microscope, the growth of fertilized embryo need to be observed in different stages like stage 1 to stage 7. So transfer of embryos is also done using non-surgical method nowadays. Because the surgical method is very invasive. Then cryopreservation of the embryos is another thing under the liquid nitrogen, and before transfer they are thawed. then this will surely make the genetic improvement and planned mating, and sometimes the disease resistant embryos can be provided, and many times the fertility may be increased, and many more technologies of the, especially the biotechnology, are there in the animal and also in the human beings, and we need to study much more. Of course, it has got just 30 to 40 years of the history, and much more research has to be done in the biotechnology as one of the emerging tool nowadays. So, thanks a lot and study well. All the best. Bye.